Welcome to Bioremediation and Dr. Mickey. Today we need to talk about the biosurfactants. What is the role of biosurfactants in the bioremediation process? And also what is the properties of the biosurfactants? Also we need to talk about what is the mechanism of the biosurfactant in the bioremediation process. Also, we need to talk about what is the classification of the biosurfactants and also what is the advantage. Also, we need to talk about what are the methods used to screen the biosurfactants efficiency. So, what is the biosurfactant will be explained shortly. Welcome back. So what is the biosurfactant? Actually, biosurfactant is the chemical compound produced by the microorganism and required to overcome the surface tension. Actually, the biosurfactant, this is produced by the microorganism as any metabolites. You know, the microorganism able to produce enzymes, able to produce the antibiotics, able to produce acids, able to produce proteins, also able to produce the biosurfactant compounds as well. So the biosurfactant is one of the metabolites of the microorganism. Very simple example, if you have, for example, the water and the oil. Water and oil cannot mix together, right? So in such a case, if you need to mix or dissolve the oil in the water, you need to add the biosurfactant. So this biosurfactant able to, overcome the surface tension between the oil and the water. So for example, the diesel oil needs to be made available to the native microorganism to stimulate the bioremediation or biodegradation rate. Therefore, the transfer from the diesel oil to the aqueous phase means you can dissolve the diesel oil in the water. Therefore, the transfer from diesel oil to the aqueous phase in the water, for example, is the key process to increase the bioavailability. So if available to the microorganisms, that means you can able to stimulate the biodegradation process. In such a case, you can promoting the biodegradation rate. So the use of the surfactant has been described as a promising method to overcome the problem of the limited bioavailability. So the presence of the surfactants, this is able to solve this problem. For example, the oil only available on the surface, water down. No, we need to be the water will be available all layers, all this area, for example. So you need to add the surfactant. So we have surfactants, we have biosurfactants. Surfactant, this is the chemical substance, but biosurfactant, this is the surfactant produced by the microorganism. So what is the properties of the surfactants? Actually, as mentioned before, this is the chemical substance containing a tail and head. The tail called hydrophobic. Hydrophobic means cannot dissolve in water. And the head called the hydrophilic. Hydrophilic means likes water, means able to dissolve in water, or called soluble in water. Here this figure shows the surfactant containing the head and the tail. The tail hydrophobic, the head hydrophilic. So if you look at this figure, you can find the surfactant containing the hydrophobic part, this is the tail, and the hydrophilic part, this is the head. And the hydrophilic part containing the sugars, amino acids, or other polar groups as well. But the hydrophobic part, this is the typically mostly aliphatic hydrocarbon, and also may be containing the beta-hydroxy fatty acids as well. So the importance of the biosurfactant, for example, if you have soil contaminated with the hydrocarbon, for example, maybe polluted soil with the hydrocarbon, so oil and the water in the soil, so the microorganism able to produce the biosurfactants to make these pollutants or these hydrocarbons available for the microorganism, at the same time to enhance the biodegradation rate as well. So now, what are the mechanism of the biosurfactants or the surfactants compounds? We have three main different mechanisms for the biosurfactant. To increase the bioavailability of the hydrophobic compounds or organic compounds in the polluted area. So able to convert the hydrophobic organic compounds to hydrophilic organic compounds. So the first mechanism, the surfactants can decrease the interfacial tension between the aqueous and the non-aqueous phase, this resulting in the formation of the micro-emulsions. So the micro-emulsions, this is when you add, for example, the surfactant to oil and water, or when the oil be available in all the container or in all the tube, you can find very tiny oil drops. This structure called the emulsion. So in such a case called micro-emulsion. The second mechanism is the enhancement of the solubility of the hydrophobic compound by the presence of the micelles. And this is the micelles looks like the aggregated particles. These aggregated particles are called the micelles. And the third mechanism is the surfactant will facilitate the transport of the pollutant. So these are the three different mechanisms of the surfactant. If you look at this figure, you can find the different surfactant, the aggregation, or what we call it the micelle. So these micelles or this aggregation in different forms. Here you can find up left, this is the surfactant, this is the monomers, no aggregations in monomers. So the polar solvent aggregation in the form of the sphericals, or maybe in the cylindrical form, or maybe in the form of the bilayer, or maybe in the form of the hexagon phase, or maybe reverse micelles. And you can find this is the surfactant monomers, another figure down. You can find the surfactant tail and the head, and the micelle will be produced as in the figure. So what is the biosurfactant? As mentioned before, the biosurfactant is the chemical substance produced by the microorganism to reduce the work required to overcome the surface tension. So the biosurfactants may be cell wall associated or may be excreted into the surrounding media. So the excreted surfactant causes the emulsification, called emulsion, of the hydrocarbons in the solution. While the cell wall associated surfactants, this is facilitates the hydrocarbon uptake. So now, what are these biosurfactant classifications? Actually, these classifications divided, or they can be classified into the biosurfactant class, producer microorganism, what is the source of these biosurfactants, which microorganism can be produced this type of the biosurfactant class, and what are the applications, or what are the different applications for these biosurfactants produced by the different microorganisms. So here, if you look at this table, I can summarize all of the biosurfactant classes and also the applications. If you look at this table, the table shows the classification of biosurfactants and their use in remediation of the hydrocarbon contaminated sites. You can find three columns. The first column, this is the biosurfactant class. This is we have different classes like the rhamnolipids, sulforolipids, allosan, liposan, and so on. So the second column containing the microorganism producing the class. For example, the rhamnolipids, this is produced by the different types of bacteria called, this is the Pseudomonas aeruginosa or some other Pseudomonas species. And the application of these rhamnolipids, this is the to enhancement of the degradation and dispersion of the different classes of the hydrocarbons. They are also able to make for the emulsification of the hydrocarbons and also the vegetable oils as well. So you can go through this table and find what is the class, what is the microorganism producing, and also what are the application. So for this table you have to know what is the different classes of biosurfactant you have, and what are the producing microorganism, and also what are the application of that biosurfactant class. So for you, no need to memorize all the microorganism, but you need to know at least one organism producing that biosurfactant. So now let's move to what is the advantages of the biosurfactants over the traditional chemical surfactants. That means, what is the advantage of biosurfactant over the surfactant? The biosurfactant produced by microorganism over the surfactant or chemical surfactants. Actually, we have many different advantages of the biosurfactants over the chemical surfactants. Here we can mention some of these advantages. The first advantage, this is the resistance to environmental change. I mean, the biosurfactant is stable at the high pH or lower pH, even between 5.5 pH or pH up to 12 or more. Also stable at high salinity from 0 to 30% sodium chloride, for example, and also stable at lower and high temperature up to 120 degrees as well, can be worse. Also, the advantage is the application and the cost effectiveness as well. So application a lot and the cost is very cheap compared with the chemical surfactants. So the production through the microbial activities makes them inexpensive, because this is already produced automatically as byproduct used by the microorganism. Also, the biosurfactants can be synthesized by using renewable resources such as molasses and sugar, while the petroleum resources used to produce chemical surfactants are limited and non-renewable as well. Also, the advantage of the biosurfactant are also biodegradability, so able to biodegradable. If the biodegradable means this is the environmental friendly as well. Also, availability of raw materials. Biosurfactants can be produced from the very cheap raw materials. Also, acceptable production economics. Depending upon the application, the biosurfactants can also be produced from industrial waste and byproducts as well, and this is the particular interest for the bulk production. So the production is the economics as well. Also, one of the biosurfactant advantages is the specificity as well. So all the biosurfactants, complex molecules with specific functional groups. So containing the functional groups are often specific in their action. So this group, this is the responsible for the action of the biosurfactants. Also, biosurfactants are the biocompatibility and also the digestibility as well, which allow their application in cosmetics, for example, also pharmaceuticals, and as functional food additives as well. Finally, the biosurfactants has low toxicity compared with the chemical surfactants. Most of the chemical surfactants are produced from petroleum, so we cannot avoid their toxicity compared with the biosurfactant. If you look at this figure shows the mechanism of biosurfactant activity in metal contaminated soil. You can find up small black dot. This is the, for example, any heavy metal. This is the contaminated in the soil, for example. This metal already absorbed to the soil surface. After that, the microorganism in the soil able to produce the biosurfactant. So sorption of the biosurfactant molecules at the interface between the soil and water and the metal complexation. So this metal complexation, after that doing the desorption of the metal biosurfactant complexes from the soil matrix to the soil solution and incorporation of the metal into the micelles. So you can find the micelles, the second last row or figure. You can find this is the micelles surrounding the heavy metal. After that you can precipitate the biosurfactant out of the complex, so you can release the metal from the soil. Now let's move to the methods used to screen the biosurfactant efficiency. Actually, we have many different methods we can do in the lab to check if this microorganism able to produce biosurfactant or not. So the first test we have or method we have, this is called the hemolytic activity on the blood agar media. That means we need to check if the microorganism able to produce the biosurfactant or not after incubation and culture on the blood agar media. So in such a case, the isolated strain streaked on the blood agar, after incubation we can find the halo zone around the bacterial colonies. This indicates the positive results for the biosurfactants production. Another test called the drop collapsing test. This is the isolated strains for the you need to check producing biosurfactant or not, placed on the surface of the hydrocarbon. So you can find the destabilizations of the cell-free broth or the broth media. Drop indicates positive results. A drop of water A acts as a control. If you look at this figure, you can find A. This is the drop of water. You can find this is the more convex. While the B, that means this indicates positive results. So that means this bacteria able to produce the biosurfactants. Also, we have another test we can do in the lab, oil spreading test. We can do this test on the empty petri dish. Two different layers were formed. The first layer would be the water and the second will be the hydrocarbon. So you can inoculate 24 hours the bacterial cell-free extract broth. After incubation, you can find the clear zone around the culture indicates the positive result. So plates B, C, and D, you can find the clear zone around the culture. This indicates positive results. That means this organism is able to produce the biosurfactants. Also, we have another test called the emulsification index test. This test in the test tube, 2 ml of the hydrocarbon you can add with the 2 ml of the bacterial culture broth. So you can mix using the vortex for 2 minutes and allow to stand around 24 hours. After 24 hours you can find in the incubation the emulsification index was calculated using this equation. Also, we have another test called the CTAB agar plate method. This is the agar plate containing the methylene blue. For this reason, you can find this plate blue in color. And CTAB, this is the cetyl trimethyl ammonium bromide. And you can inoculate the bacterial culture and incubate for the 24 hours. So you can find this dark blue halo around the bacterial colonies as a plate. This is the biosurfactant production is positive. The last method we have, this is the hydrocarbon overlay agar method. This method, we can inoculate the bacteria and incubate for the 48 hours. After incubation, the growth was observed on the LB plate. This is the plate media. and the colony surrounding by the emulsified halos was considered as positive for the biosurfactant production. Like this picture, we have A, you can find the A number 1, this is control, no emulsified halos, but number B, you can find the emulsified halos zone. This is the considered positive biosurfactant production. So these are some of the methods to screen the biosurfactant efficiency or the production of the biosurfactant in different media. So this is the end of this video, and I hope you get the benefit, and please don't forget to share, like and subscribe and activate the bell to reach all of my new videos. Thank you, good luck, and bye bye.